So the photosynthesis, okay. So during World War II, Kelvin used C4 radioactive isotope technique, okay, by alga, okay. They found out C3 pathway, okay. Then photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Okay. All all living forms on Earth depend on sunlight for energy. Okay. Two reasons photosynthesis is very important. First, it's the primary source of all food on Earth. Okay. It is also a release of oxygen. Now, what do we know? Like, um, what is required for photosynthesis? Chlorophyll, that is green pigment of the leaf, then light and CO2. Okay. Now there is experiment of variegated leaf. Okay. Now on testing these leaves for the presence of starch, it was clear that photosynthesis occur only in the green parts of the leaves in the presence of light. Okay. Now another experiment, that is KOH soak cotton which absorbs CO2. It showed that CO2 was required for photosynthesis, right? Now early experiments, Joseph Priestley discovered the air essential on bell jar experiment given by Joseph Priestley, and Priestley hypothesis that plants restore to the air whatever breathing animals and burning candles remove. Okay, now Jan Engine House. What Jan Engine House did? He showed that it is only the green part of the plants that could release oxygen. Only green parts of the plant that could release the oxygen, right? So this is the Jan Engine House stated. Okay. Now in 1854, Julian Vances provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. Okay. Now Joseph Priestley found that the green parts in plants is where glucose is made, and that the glucose is usually stored as starch. So green part where the glucose is made, and it is usually stored as starch in plant, right? Now Engelmann, what he did? He take green alga, that is Cladophora, in suspension of aerobic bacteria, that is Azotobacter. Now the bacteria detects the site of O2 evolution, right? Now he observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. Okay. So a first action spectrum of photosynthesis was thus described by the T.W. Engelmann, and this spectrum, that is red and blue light, resembles roughly the absorption spectrum of chlorophyll A and B. CO2 and H2O, in presence of light it forms CH2O and O2. Okay, CH2O is carbohydrate. Okay, now Cornelius van Neel studies on purple and green bacteria, that he showed that the hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. So H2A plus carbon dioxide. So hydrogen from H2A, it reduces CO2, that is CH2O, and I mean H2A gets oxidized to A and H2O. Now H2 is the hydrogen donor and is oxidized to O2. So water in photosynthesis is oxidized to oxygen. Now when we take the H2S, okay, there is formation of in for that the O2 evolved by the green plant comes from the H2O, not from the carbon dioxide. So this is what Carnal Swan Neil, and then he also take like radio isotope technique to prove that O2 release is from water, not from the CO2. Photosynthesis is not a single step reaction but distribution of multiple step process. This is photosynthesis. Now where does photosynthesis takes place? In chloroplast, right? So mesophyll cells, okay, they have large number of chloroplasts. Now chloroplasts usually align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. Now the membranous system consisting of grana, the stroma, lamella and the matrix stroma. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. ATP and NADPH, right? So in stroma, enzymatic reactions synthesize sugar in stroma, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reactions, that is photochemical reactions. The latter one, they are not directly light driven but are dependent on the products of light reactions, that is ATP and NADPH. So these are like dark reactions, that is carbon reactions. That doesn't mean they are not light dependent, they are dependent on light but they are indirectly depending. Okay, outer membrane, inner membrane, there is stroma, lamella, then granule thylakoid. Okay, you see like stroma, lamella, then grana, then stroma, then ribosomes, then starch granule, then lipid droplet, right? Now you see how many types of pigment are involved in photosynthesis. So major pigments are chlorophyll. Chlorophyll show the color like bright or blue green. Okay, bright or blue green in chromatogram. Then chlorophyll be yellow to green. Bright or blue green, bright chlorophyll A. In chlorophyll be yellow to what? Yellow to green. Then xanthophyls, it is yellow. Carotenoids, yellow to yellow orange. Now the maximum photosynthesis. Okay, maximum photosynthesis occurs in plant. Okay, the wavelength at which there is a maximum absorption by chlorophyll A, that is in the blue and red regions. The blue and red region also shows higher rate of photosynthesis. Okay, so and you see like there is complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis. You see the photosynthesis maximum in the absorption spectrum of chlorophyll A and B, which absorbs red and blue light, right? First you are showing like chlorophyll A, then chlorophyll B, and then carotenoid, right? Like rate of photosynthesis measured by O2 released, then light absorbed. Okay, so graph showing action spectrum of photosynthesis superimposed on absorption spectrum of chlorophyll A. And then what is light reaction? There is like P700, P680, P700. Uh, it has chlorophyll A has absorption peak at 700 nanometer wavelength. So this makes the complex, that is PS1 complex. Okay, now P680, it has a reaction center of wavelength, that is 680 nanometer. It is PS2. Now PS1 is present in both PS1 and PS no both in thalate membrane of the thylakoids, that is granule thylakoids and stroma lamella. So both have the PS1, but PS2 is absent in stroma lamella. Okay, and also NADP reductase is also absent in PS2. And so there is like electron transport. There is like electron transport, that is ETS. Now the electrons are picked up by an electron receptor which passes them to an electron transport system consisting of cytochromes. So electron transport system consists of the cytochromes, right? Now this represents the Z scheme. Z scheme, it is non-cyclic photophosphorylation. Phosphorylation is the formation of the ATP. Now non-cyclic photophosphorylation products are like ATP and NADPH. NADPH, right? Now water splitting associated with the PS2 on the inner side of the lumen of the thylakoid. H2 gets converted into 2 electron plus 2H plus oxygen. Oxygen gets released later, but the electron get passed on to the electron acceptor. Okay, now this is the Z scheme, and yeah, so this is the Z scheme. And there is, and on the stroma side there is NADP reductase, that NADP plus gets reduced to NADPH. Okay, now splitting of water uh, inside the lumen, H plus gets increased, and this is how they create a proton gradient across the thylakoid membrane. So when the proton passes through the thylakoid membrane, you see thylakoid membrane has like channels, that is ATP synthesis at two channels, that is CF node that embedded in the membrane and CF1 at the periphery. Okay, so when hydrogen passes through it, there is formation of ATP, and when the electron of water accepted by the PS2, that is P680 reaction center of chlorophyll A, then it passes electron to plastoquinone, then cytochrome P6 complex, then plastocyanin, then PS1, that is P700, then FD and FNR. Okay, FD and then FNR. As you can see, so in the overall stroma side gets lesser hydrogen, and that builds up the proton gradient across them. Okay, now within the chloroplast proton. Okay, chemiosmosis, that is formation of the ATP, requires a membrane, that is thylakoid membrane, a proton pump, so that from the stroma side we can transfer the hydrogen ion into the lumen to maintain the proton gradient, then proton gradient and ATP synthesis. So immediately NADP and ATP used immediately in the biosynthetic reactions. Okay, and biosynthetic reaction is responsible for fixing CO2 and synthesis of sugars, and then it is stored as starch. Now, what are the ATP and NADPH used? Where are the ATP and NADPH used? In here, cyclic and non-cyclic. As I told you, like in cyclic there is only ATP formation, okay, because same electron gets recycled back to them. But in PS2, the electron is lost, so it is maintained by the water, okay. And I also told you, like while the membrane or lamella of the grana have both PS1 and PS2, the stroma lamella membrane lack PS2 as well as NADP reductase enzyme, okay. Okay, the cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H plus, okay. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation, right? And also ATP synthesized in the chloroplast. This is chemoosmotic hypothesis, okay. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the ETS. So between the two membranes, okay, there is accumulation of the proton. So where are the ATP and NADPH used? So ATP, NADPH and O2, these are the products of light reaction. ATP, NADPH and O2. So O2 diffuses out and ATP and NADPH used in biosynthetic phase. Now this process does not directly depend on the presence of light but is dependent on the products of light reaction, that ATP and NADPH besides CO2 and H2O. Okay, there is dark reaction now. So during World War II, Melvin Kelvin used radioactive C40 in algal photosynthesis. Okay, uh, this leading to the discovery of first CO2 fixation product, that is 3 carbon organic, and it was 3 carbon out, and this was